Hey guys, so this video is just to introduce what Punnett squares are and what we use them for, what they are, okay? And what we use Punnett squares for is just to predict what type of offsprings you would get if you cross one organism 1 with organism 2. So if we could be talking about flowers, we can be talking about uh, plant, any type of plant, any type of tree, really any organism, right? If we can just be looking at, or if we're just looking at one trait or two traits, more than two traits, you can do it as well, but it would just be a huge Punnett square. Some of you guys are used to, or are used to the term Punnett square, or have seen Punnett squares that work with them. Some of you guys have not. So I will introduce what they are, what we use them for, and how we actually solve these types of problems, or how to predict, okay, what type of offspring we would get from two organisms. And in order to do that, we need to use these four rules. Okay, we'll talk more about them in class, but the first one is we gotta determine the alleles of the parent. The alleles, we know that are letters, right? And every gene or every trait, okay, is represented by two letters, a big letter and a little letter, or it could be two big letters or two little letters, right? We know that the capital letter is always dominant over the recessive or the little letter, okay? But again, every trait, black hair, your color hair, the color of your skin, the color of your eyes, all of that is represented by two letters, okay? Or when we're talking about genes. So, and that's the first thing we need to do when we're talking about, when we're solving Punnett squares. So we need to determine the alleles of the parent. Then we're going to determine what type of gametes that parent can give. So if we have, what that means, just to be go through it fast, we know that, let's say that dad is a big B, little b, right? And he represents black hair. So the gametes that he can give to his offspring, because remember, meiosis cuts everything in half. So the gametes that he can provide are a big B or a little b. Okay, so we need to determine that. Once we do that, then we can find the combinations. We make our Punnett square, and we can find the combinations of how many offspring or what offsprings would look like. After that, then we can determine different types of ratios, and the ones we're going to be talking about is genotypic ratio and phenotypic ratio. Okay, so let's talk about monohybrid crosses. Monohybrid crosses are the ones that you're probably more familiar with if you work with Punnett squares, and that's the Punnett squares or crosses that only require four squares. Okay, it's a very simple cross, and it's just looking at one trait. So you're going to be looking at a flower, the color of a flower, the color of hair, the height of a plant, right? And so you're only going to be messing with one trait or one characteristic of that organism. So let's just do an example real quick. Okay, let's say that we're going to have, let me set this up real quick. Let's say that we have a plant, and that plant we have could either be red, so big R is for red, or little r is for white. Okay, and we're talking about Mendelian genetics, so it's either going to be red or white, right? And we are going to look at, and this is only one trait, so this is one trait. So we know that we're going to be looking at a monohybrid, and it's one trait because the only thing we're looking at is the color of the flower, right? And let's say that parent 1 is going to be a big R, little r, which is a heterozygous red, and we're going to cross that with parent number 2, which is going to be homozygous dominant red, right? So two big R's. So we know we're looking at one trait. So the way we're going to do this is we're going to set up our Punnett square, right? We're ready, and we know we're using this type of square because it's one trait. Okay, so let me move this real quick. We'll put it over here. So now if we go through step 1 is determine the alleles. Well, we know parent 1 could only give an R, big R, or a little r. So we can put it over here. Okay, we know every square is for one allele, and you can put the big R, little r on the top. It doesn't matter where you put it as long as you keep it together. Parent 2 can give a big R, or it can give another big R. So we put that right there. Okay, So that's what the alleles or the gametes that they can give. Okay. Once we know the alleles of the parents, we know what gametes they can give. Then we can do our Punnett square. So we're gonna cross this one with this one, and that's gonna give us big R, big R. This one is big R, big R. This one is big R, little r, and then this other one would be a big R, little r. Okay. And it doesn't have to be big R, little r. You can have little r, big R, but it's always easier to identify what type of organism you're looking at, or the genotype or phenotype of these organisms, if you put the capital letter in front. Okay, so that's just a quick introduction of how we would make a monohybrid cross. And the other type of crosses that we're going to be looking at are going to be dihybrid. And in dihybrid crosses, we're actually looking at two traits. Okay, so two traits. And these crosses are going to be a little bit more complex, because you're going to have a 4x4 cross, which is going to equal to 16 offspring. Okay, so we're going to go through one real quick, and then we'll go into more detail in class. Okay, this is just a quick introduction again. So let's say that we're looking at the color of feathers. Okay, so this trait right here, a big G or little g, tells us if it's green feathers or yellow feathers. Okay, and then the other one, okay, the other trait that we're looking at is going to be the size of the beak. Big L is long beak, little l is short beak. We're not actually going to do the Punnett square today, but I'm going to show you how to figure out the alleles and the genotypes. Okay, so let's say that parent 1, okay, so parent 1, it has it's homozygous dominant for green feathers. So what that would give us is big G, and then is heterozygous for the beak. And we know that heterozygous would be that. Okay, so that's parent 1. Okay, parent number 2, let's say that is heterozygous for both traits. So that means he's heterozygous for the color of feathers, and he's also heterozygous for the size of the beak. Okay, so that's the first step. Okay, the first step, remember, that was determine the alleles of the parents. We know parent 1, sorry I didn't put parent number 2 here. Parent number 2 and number 1, we already know their alleles. So number 2, the step number 2 is to figure out the gametes. What are the gametes that organism can give? And this is the difference between monohybrid and dihybrid. Okay, remember that in monohybrid, we're only looking at one trait. So if dad was like that, the only options was to give a big B or a little b. Okay, so that was the options on monohybrids. On dihybrids, we're actually going to do something that you guys have done in math, and it's called the FOIL method. So we're going to bring this down, okay, and then we're going to use the FOIL method, and we're going to combine these alleles so that we can end up with 4 types of gametes. So we're going to have big G, big L, so that's one gamete that parent can give. Outside, inside, outside, outside. So now we're going to go outside, outside, and that gives us big G, little l. The other way is, and then we're going to determine the other one, right? So we're done with that outside G. Now we go with the inside G. Inside, inside, so that's GL, GL, and inside, outside. So that's big G, little l. So those are the 4 types of gametes that the parent 1 can give. Parent 2, okay, I'm not going to rewrite it, but parent 2 would give us, again, outside, inside, right? So it would be big G, big L, outside, outside, so big G, little l, inside, inside, little g, big L, inside, outside, and that gives us little g, little l. So now we have step number 2 completed, and we know that that's what these 2 parents would give to their kids. Okay, they would give those 4 types of gametes. Okay, those are the possibilities. So if I shrink this over here, okay, I'm just going to put it right there, and we're not going to complete the Punnett square, but I'm just going to show you if we were to set it up. Remember that when you're looking at 2 traits, okay, you're gonna have a Punnett square that has a 4x4. So you would have 4 going down, 4 going across, Okay, and it doesn't matter where you put parent 1 or parent 2. You can put parent 1 on the top or parent 1 on the bottom. And if we decide to put parent 1 on the side, okay, so we would end up having a big G, big L, big G, little l, okay, big G, big L, big G, little l, right? Those are the 4 types of gametes. And then we put parent 2 on the top, and that would be big G, big L, big G, little l, little g, big L, little g, little l, okay? And that's how you set it up. If you were actually to cross it, right, let's say we were going to try to find the offspring of or the genotype of the offspring in the X. So we would actually go across, just like we did in monohybrids, right? So we would go across, okay, and then we'll grab the G's first. So we know we have a big G, we know we have a little g, so we put those two because they're the same. And then we grab the L's. We have a big L and we have a little l. So there you go. Your offspring would be heterozygous for both traits, and you have your 4 alleles that represent the 2 traits. Okay, so that's just a quick introduction on how we do this. We're going to do a couple more in class, and then you're going to get to practice some more. But it's just to introduce it so that when we get to class you can bring some questions, and while we're doing it you can understand it even better. Okay, all right, have an awesome weekend.